Okay, so our next speaker is Jyothish Soman. He's Director of Engineering at Relation Therapeutics. This startup was the power. This startup uses the power of recommender systems, machine learning, and large-scale, high-quality data that describes disease biology to find better ways of targeting pathology and treating patients. He was previously a postdoc at the University of Cambridge, where he also did his PhD. He has spent multiple years in academia and industry, researching topics ranging from HPC, graph analytics, knowledge graphs, graph ML, and fault tolerance. So today he'll discuss utilizing graph machine learning within drug discovery and development. Okay, on that, I'll hand over to you.
Hi, everybody. So glad this got sorted. My name is Jyothish Soman. I am the Director of Engineering at Relation Therapeutics. And thanks for taking out your time to come and listen to me talk about graph-based machine learning. And next slide. So in this talk, we're going to be talking about the major attention regions for machine learning and drug discovery, and especially the regions of interest when it comes to us. And then we are going to be slowly moving into graph neural networks, a slight small introduction into the domain. So one of the focus regions of this talk is going to be GNNs and molecule design. And one of the other parts is obviously drug repurposing and combination repurposing. And I'll finish off with a small introduction into Recover, which is one of the projects we've run with Mila for the last year. Next slide, please. So before going further, I would like to obviously introduce Relation Therapeutics to everybody. So we would like to call ourselves pioneers in recommendation systems in biology, in aiding drug discovery as well as development of both stages of the pipeline we are focusing on. But right now, our primary focus is on the discovery side of things. So to attain that, to build this recommendation system, what we use is graph-based systems. So to really understand the biology and accelerate the process. To do so, we use multiple different data sources, all the way from human genetics, single cell profiles, functional genomics, and we also try to generate internally a high confidence causal relationship map, which drives diseases. Next slide, please. So this is one of our favorite diagrams over here, where we are discussing the attention regions for machine learning. So as you've seen, we've basically spread out the entire pipeline for drug discovery along two axes. One is the cost involved, and the other one is the number of programs that run. And in each stage of this drug development and discovery pipeline, what are the regions in which machine learning can really help? For example, the first one is obviously target identification, which comes right at the beginning of the pipeline, and then comes in design of small molecules, high throughput screens, enhancing them, de novo design, property prediction, and then obviously biologics, where we go for de novo design, protein engineering. So quite a few things in the drug discovery part of things. And once we move into the drug development, one of the primary focuses for us, primary interest area is drug repurposing. And we look at drug repurposing across three different lines. One is off-target repurposing, on-target repurposing, and combinational repurposing. So that's been one of the focus areas for us. And obviously there are other trajectories for machine learning and drug discovery. For example, the clinical trial design, biomarker discovery, and looking at real-world evidence and electronic medical records, personalized medicine. So there's a few aspects of interest. One of the aspects we are really concerned about is that while drugs move through this really large pipeline, which generally take like 10 to 15 years, the attrition rate, the rate at which drugs actually fail is significant. For example, if we start off with 25 programs, you're more than likely to end up with one, and the cost slowly and steadily ramps up over time. At the end of the pipeline, you're looking at greater than $1 billion spent on bringing one drug to the market. So our proposition is that using machine learning in drug discovery across all of these different phases, if we can actually improve efficiency by a significant bit in each stages of the pipeline, two things happen. The cost of drug discovery goes down significantly, and the other one is that efficiency of the pipeline improves as well. So that is one of the primary drivers of our work. Next slide, please. So this is again a rework of just a listing of what I've talked about earlier, the attention regions for machine learning and drug discovery. In this particular conversation, we're probably going to be talking a bit about small molecule design and drug repurposing, and less about the other ones. Next slide, please. So, as I mentioned before, this talk is about graph machine learning, and we cannot actually have a conversation on graph machine learning nowadays without actually having a discussion on GNNs. So, so before going into a deeper discussion on GNNs, I just want to introduce GNN to the larger audience, just to make sure that everybody understands what I'm going to be talking about. So GNNs are basically machine learning that runs on graphs. So neural networks specifically. So typically a graph is represented as a collection of vertices, edges, node features, and edge features. And what GNN does is like a typical neural network. Each particular layer takes in the input features of the various elements. For example, if it's an image, it looks at the n by n into 3 matrix. In this particular case, it'll be looking at this particular graph to generate the next set of representation. And in this particular case, we are looking at a graph which would be looking at its immediate neighborhood. So what it's going to be doing is that it's going to be looking at its neighbors, both edges as well as nodes, and it's going to be accumulating messages from the different nodes in the system. Sometimes the edge features are also considered, and a function is ran on the node features and node features of the neighborhood as well as the node of its own. And based on that, the next representation of each individual node is actually considered. So that's a typical layer of a GNN. There are obviously variants which do much more complicated things, like look at a much larger neighborhood, or look at the subgraph within which the node actually belongs in, or do graph pooling, where a collection of nodes are agglomerated into a single node. So, so there are variants of it, but a simplistic view of GNNs is that every node looks at its immediate one-hop neighborhood and does a functional a function is ran on those features to figure out what is the next node representation. So that's the simple plain vanilla explanation. So next slide, please. So one of the most simplest examples is of a GCN. So I understand that earlier I was discussing on a per node basis. So GCNs are slightly more easy to understand as a matrix, three set of matrices being multiplied to each other to find the next representation. For example, here H of L+1 is the next set of representation for the entire for all the nodes in the graph, and that is driven by the weighted Laplace, the normalized Laplacian, multiplied by the current node representation given by H of L, and the weight assigned to that particular layer of the GNN. For example, if there are like 4 layers and the 4th layer the output of the fourth layer h of 4 would be equal to the sigmoid of sigmoid multiplied by sigmoid of the Laplacian of the graph multiplied by h of 3 into weight of the fourth weight of that particular layer. So so this is a rather simple example, and if we take out the sigmoid, it just becomes it becomes a power of it becomes the weights of the individual layers being multiplied with each other. So it becomes w pi of w into h of 0 into the Laplacian powered up. So so that's why I find it a bit simplistic. Next slide, please. So there are GCNs are a simplistic version, but nowadays we are going into more and more complicated architectures. So we are going into geometric deep learning, geometric GNNs. But before going into that, there are GATs, which is basically attention-based graph neural networks, and MPNNs, which is more like a framework for doing message passing. MPNs are fairly similar to the definition we talked about earlier, where we define for each edge a function, for each pair of nodes, as in node A, node B, and there's an edge in between. So there is going to be a message being passed from node A to node B, and that is going to be defined by the current representation of node A, node B, and the edge features. So those three put together would define the message moving between node A and node B. And the next set of representation of a single node is going to be, it's going to look at all the messages that are coming in from its neighborhood, and it's going to do invariant function, for example, summation on all these messages that are coming in, and that summed up value and the current representation is used to generate the next representation. So basically h of L plus 1 that happened in GCN based on h of L, a similar thing is going to be happening, but based on messages instead of the Laplacian and the weight matrix. So that is the message passing networks, and then comes a slightly more complicated, not complicated, as in it's fairly similar to the transformer attention models that we all are very much familiar with. So so there's a attention layer where each pair of elements in a transformer actually realize how much of an attention they should be giving to each other. But this is on a graph, so it's not all to all. So the attention is only on nodes which are connected to each other. So an attention function is used to generate that particular attention value between two different nodes, and then a softmax is run on it on that particular value, exactly like transformers, and then eventually a sigmoid function on that sigmoid function is run exactly like a transformer would be happening. So that's the graph attention module. So these are the two fairly common GNN models that are in practice. Next slide. So there's been a lot of, so beyond drug discovery, there's been a lot of other application areas for GNNs, which include traffic forecasting, knowledge graph completion, learning on incomplete graphs, recommendation systems, combinatorial optimization. And one of the very useful applications which I've found is chip placement, which for VLSI and EDA firms is very, very important, and it's a combinatorially explosive space. So it's a very interesting application. That one is specifically something I really enjoy. Next slide, please. So that was other domains, but now GNNs are becoming very much ubiquitous in bioinformatics nowadays. So the field is really exploding. So everywhere you look, you'll be able to find GNNs being applied to every possible problem that is there. So there's a lot of interest that is getting generated. The bioinformatics field is growing exponentially right now, and so is the field of graph neural networks in bioinformatics. So there's obviously pushback from and reservations in the community regarding this. For example, we have SPM and XGBoost, which are really amazing at explainability, and they can really work with smaller data sets, and which are typically not something GNNs are really great at. So there are certain aspects of there are certain methods of machine learning which are still quite useful and being used. So I'm not saying GNNs are the panacea and they'll solve everything, but they definitely are able to do a lot more stuff. And especially as the amount of data that we have, network data that we have, is growing exponentially, it's a very good space to be in. Next slide, please. So now coming back to areas of interest. So so this talk is about largely about molecules. So one of the major areas of interest for graph neural networks in bioinformatics is molecules. And interestingly, it's very simple to actually represent a molecule as a graph. So each of the atoms becomes a node, and each of the bonds becomes an edge. There are variations to it. So if two nodes are two atoms are close by in a physical sense, there is a different type of edges that can be added, but you're not going into that. We have, in a simplistic sense, if two atoms have bond with each other in the molecule, we consider them to have an edge in the graph. And one of the important aspects of such graphs is that they are planar. And the planarity of a graph is very important because, can you move to the next slide, please? So planarity is a very important concept because one of the things you would constantly find in graph neural networks is the WL limitations. So basically, most of the graph neural networks can be equated to a form of the Versifella-Lemon test, some order of it. So there's a bound on how much an expressive neural network can be. WL is basically a graph isomorphism test. So, but for planar graphs, there are O of n square algorithm available, which are practically able to find if two graphs are isomorphic. The best solution is in order of n, but that's not a very practical algorithm. So, but yeah, so it's still a polynomial algorithm which is available for actually finding planarity of small graphs, and especially molecular graphs that we are dealing with are fairly planar. So, so expressivity of the GNNs that are specialized for molecules is going to be pretty good, and having very domain-specific models which deal with planar graphs and molecules is going to give us much more expressivity than plain vanilla GNNs. Next slide, please. So generally the setting for GNNs with molecules is inductive, where we are looking at different graphs and how they match up. So a lot of issues can be papered over. For example, homophily and heterophilia are not, while you're designing a GNN, you're not actually being worried about it. And exploitation of the structure of the molecule is primary driver, like junction tree and gemnets are very good examples where the structure of a graph is properly utilized in generating insights. And slowly we have geometric methods which are coming up, which are becoming really popular, like ICLR22, which is going on right now. There's a paper, GeoDiff, by Jian Tang, who's actually one of our advisors, which has just come out. So it's a very interesting space right now. Next slide, please. So gemnets, as I mentioned before, this is an example of a very specialized GNN for molecules. So as I was mentioning before, expressivity of a neural network. So basically how much information it can actually infer about the graph, that is very important. So with gemnets, one of the aspects they've done is they've looked at the representation and model expressivity, and they do so by looking at the geometric version of the GNN, and they look at spherical representation for edges. So instead of working in the Cartesian coordinate, they're working in the spherical coordinates. And instead of using something like Fourier transform, they're using the spherical Fourier Bessel transforms. And that is providing competitive results. Basically, the suggestion is that domain-specific graph neural networks are a very tempting region to be in. Next slide, please. So before moving further, so we've been talking about molecules, but we also need to be having a slightly broader view of what are the different data sources for machine ML-driven drug discovery. Obviously, we have drug combinations, drugs, we have proteins, both structure and sequence, and we have protein and PPI, the protein-protein interaction graphs, we have gene interaction graphs, we have gene annotations, we have diseases, we have sequencing data like TAG-seq, ChIP-seq, and so on. So there is a lot more information and data sources which is coming into this particular domain, and that makes it both interesting as well as challenging. Next slide, please. So one of the areas of interest for us is drug repurposing. There are different kinds of repurposing. One is on-target repurposing, where what we do is we look at the drug target and genes associated with a particular disease. And we call it on-target because we are still targeting the same same drug target as the as has been previously published, but we would ideally be looking at it in a different context. For example, we'll be looking at targeting a different tissue type. So that is on-target repurposing, and the other one is off-target repurposing, where the drug is now targeting a totally different protein, for example, and so that is of a really good interest. And one of the interesting things we found out is that protein structure is not really, it's a great amplifier for the results, but it's not fundamentally necessary to get some good results in. Next slide, please. So off-target repurposing, so we have a large number of proteins. We have like 21,000 proteins in humans, and there is there's obviously a need for actually inferring a lot of missing drug target interactions, and that's the entire field of off-target repurposing. So so even though there was a dearth of protein structure with AlphaFold coming into the picture and similar architectures, there is now access to a lot of protein structure, which has accelerated the process of drug repurposing. So which was earlier heavily reliant on sequence-based methods. Next slide, please. And one of the important fields of work is actually getting good protein embeddings, because the sequence tends to be large. So so the first one obviously is protein structure-based embeddings, where it's very useful to use geometric deep learning. And to be honest, transformers can be looked as a version as a geometric deep learning method. And the GNN, it's a bit of a simplification, but transformers can be looked as versions of GNNs. And then we have sequence-based embeddings. We have like ESM, which was there from Facebook research, which was very useful, which was shown to actually incorporate protein structure implicitly within the embedding as well, which was very useful. So there have been a few methods for protein embeddings which can be used with for off-target repurposing. Next slide, please. So now comes combination repurposing, where the focus is now, instead of looking at a single drug and a single target, now what we're looking at is a combination of drugs. We are looking at like two drugs put together. Would they be interesting for a particular new disease? For example, what would be a combination which would be useful for COVID-19, for example? So that was part of a collaboration with Mila, the Recover project, in figuring out combination therapies for COVID-19. And the paper is out on arXiv right now. So So if you want to have a go at it, please do have it. Anyway, so the issue is that there are about 4,000 approved drugs, and just look at the combinational space that involves about 8 million experiments, and that is a humongous space, and the amount of time and resources needed for a single experiment is non-trivial. So we are looking at like millions of dollars worth of money spent on just doing millions and billions of dollars worth of money just doing an exhaustive search on this particular domain. So that's not really practical, and that's why doing combinational repurposing using machine learning is very lucrative. Next slide, please. So one of the things that was within combination therapy that we found really interesting was there was a work by Alberto Parabasi on drug repurposing on COVID-19. So they do an ensemble of methods, including network analysis and GNNs. And so what was found was ritonavir was found as a top screen by the Parabasi group. But interestingly, their screens actually failed. But a combinational therapy using ritonavir, and it's called Paxlovid, has been strongly recommended by WHO like 3 or 4 days back for COVID-19. And NHS already has been using this particular combination in hospitals. So the power of using combinational therapy is pretty good, and it is pretty potent and useful, and it is a direction in which the world is going towards, for sure. Next slide, please. So what we've been doing with Recover is that we've been building an active learning pipeline, wherein so we have trained model which would be able to predict which would be able to predict drug synergies, but which would also be able to use using an acquisition function tell us which region of which set of experiments would likely yield better understanding of the field of drug. So whereas they're the most uncertainty in the predictions, for example, and then use that to drive wet lab experiments. And then from the wet lab experiments, new training data comes in, which would guide the next round of experiments, and so on and so forth. So basically, we would be able to narrow down experiments that need to be run in the lab much more quickly. Next slide, please. So this is basically the Recover pipeline in a single slide. So as I was mentioning before, we have the machine learning pipeline on one side, and then we have the wet lab on the other side, and then an active learning loop brings both of these together to build a pipeline which is directed towards drug repurposing. And that is my presentation. And as always, we are hiring people. So if you're interested, just send me a message.
Right, Jyothish, thanks very much for that. So another really good talk. It's nice to see these sort of the machine learning and the wet lab sort of coming together, sort of validating each other as they move along.